If we focus in on the regulation of the gene for that series of enzymes to make tryptophan, okay, we get this picture here, and there's a whole lot going on in this picture, so we're going to just try to focus on certain areas. First of all, what we're calling this entire thing here, okay, and we're focusing over here, this thing is called an operon. It's called the TRP operon or the tryptophan operon, okay. An operon is, okay, I've just listed down here, an operon always consists of an operator, which is something that turns on and off a switch, or it turns the genes on and off, so we're going to call it the switch as the operator. There's a promoter region, which all genes have promoter regions. That's how, if you remember RNA polymerase, that's how it knows to start making a transcript or to start doing transcription. So there would be a promoter here that says, hey, there's a gene coming up that we should transcribe, and then RNA polymerase can attach there and start to transcribe the gene. So there's always a promoter region in an operon, and then there's the actual gene or genes that we would like to turn on or off. Okay, if you remember in the previous slide, there was 5 genes for the tryptophan pathway. And so these are those enzymes. They end up all being clumped together in one section of DNA, and they have one promoter for the whole group of them, which is really cool because if there's one promoter, that means if we're making any one of these enzymes, we're going to make all of them, which is exactly what we would want because we need all 5 of them to make tryptophan. So we can assure that they're all being produced at the same rate. Okay. If this gene is turned on, and typically operons are, if you don't do anything to them, they're going to be in the on position. If they're turned on, then our promoter is working. RNA polymerase can start making a transcript after the promoter. This operator is doing nothing. It's just kind of existing there. And then we have the E gene, the D, the C, the B, and the A gene. And if you'll notice the way we can make, even though it's one transcription unit, here, the reason we can make different genes or different proteins out of it is because there are start and stop codons when we go to translate this. So if there's a start codon here and we translate, meaning making the protein, translate until we get to a stop codon, that's going to make the one enzyme. Okay. There's another start codon here and a stop codon there, and you can see this along here, so we can end up making 5 different enzymes out of one long piece of mRNA, or one transcription factor, unit. Anyway, so that would be normal. This whole thing's in the on position, okay, the operon. Now, over here, we have something that we call a regulatory gene, and I'm going to get a different color so I can circle these things. A regulatory gene. Okay, and a regulatory gene simply makes a regulator. In this case, it's a repressor protein. Okay, and so this gene, notice it is not attached. We've got a little split here. It's not attached to the operon. It's somewhere else in the genome of the bacteria, and it codes to make a repressor. So here's our repressor protein. That repressor protein in this case is not active. When it's made, it's not active, meaning it doesn't really function for anything. Okay, it's in an inactive state. And so we have this repressor that doesn't do anything. These repressors are always, they're always present in the cell. Okay, so this would be the normal in the case where there's not any tryptophan. This is how everything would be working in the cell. Okay, we have our operon functional, it's making the enzymes that we want, and we've got repressors that have been made or are being made, but they're not active, and so they're not going to do anything to turn off this this whole system. Okay, the next slide here. This slide shows what happens if we have tryptophan present in high quantities in the cell. Okay, and if we do, if we look over here at this regulatory gene, our regulatory gene makes its transcript, which ultimately makes this repressor protein. Well, remember that's inactive, this repressive protein, unless tryptophan ends up being a regulator here, a regulatory molecule, and tryptophan will attach to the repressor, and when it attaches to it at a site that's not the active site, it makes this become an active repressor. Okay. Well, the active state form of the repressor, the active form of the repressor can attach. These aren't labeled here, but this is the operator region of our operon. Okay, so that's the operator. It can attach to the operator, and what that means is the green here is our promoter. Here is our RNA polymerase. RNA polymerase, when it goes to attach to the promoter, it can do that, so it attaches to the promoter. But when it goes to go down the line to transcribe our genes so that we can make more enzymes, it runs into this repressor, this activated repressor, and that prevents the RNA polymerase from doing its job. So over here we have the genes for the different enzymes for our making of tryptophan. Those genes, they never get transcribed, which means they never get translated, which means the enzymes don't get made. And so we have effectively turned off this gene, and now we're not making as many enzymes to make tryptophan. Okay? As tryptophan levels get lower, if you remember back to the unit where we learned about enzymes, if you remember, enzymes are always changing shape, going from the active to the inactive form. So this repressor would actually be going from active to inactive to active to inactive. And this tryptophan molecule, if it lands in there when it's in the active state, it'll hold it there so that it can do its job. But these tryptophans will often be coming back out of this, which would release this protein. If there's a whole lot of tryptophan in the cell, then it means that we're turning these genes off the majority of the time. But as tryptophan gets used up and there's less of this co-repressor, they call it, if there's less of the co-repressor to turn the repressor on, then all of a sudden this gene will be on a lot more often. Okay, and yeah, so to me that's kind of a really fascinating or neat way for things to happen. We're going to look at one other type of operon here really quickly, and I think we'll call it a video at that. So.